Number 7, new aspects of gene therapy. The brain is where many of the most pervasive diseases of the central nervous system arise, including many ailments that have changed people's lives for the worse, especially for the elderly. Some diseases are triggered by inherited genetic mutations, while others stem from injury, inflammatory processes, or genetic alterations from environmental sources. Unfortunately, available therapies, medications, and surgeries have neither cured nor effectively slowed the advance of these neurodegenerative ailments, due in part to the complexity of the brain and the blood-brain barrier in particular, which effectively blocks entry to many neurodegenerative and psychiatric therapeutics. Gene therapy was widely heralded in the early 1990s as the answer to many diseases. Improved understanding of underlying disease mechanisms, as well as improvement of therapeutic gene selection and methods of delivery, are some of the reasons for renewed enthusiasm for the success of future gene therapy efforts. Researchers are developing better vectors and more confidence in gene therapy applications as benefit is starting to be seen in phase 1 trials with no related adverse events. Thousands of clinical trials have been carried out to test the safety and, in some cases, efficacy of gene therapy in neurological ailments. Some results have been encouraging, suggesting that many different versions of this approach will be translated to the clinic in the near future. These therapies will be designed to arrest disease progression, cure and/or reverse the damaging effects of a wide range of disorders of the central nervous system. The basic principle is to use gene therapy to replace a faulty gene or else blunt or turn off a specific disease-causing mechanism. There are more than 10,000 human diseases that are caused by a single gene defect. But in the future, gene therapy should be a way to fix some of these genetic problems at their source. In addition, even diseases without a known major genetic cause, such as brain tumors, Parkinson's disease, and Alzheimer's disease, can be mollified by introducing therapeutic genes and vectors.
So we heard a wonderful session this morning on gene therapy, and we have a chance now to hear more from Dr. Sandra Brakefield about this. And Xandra, it makes sense that in the field that people have started with rare diseases of very specific mutations, known mutations. When do you think, and how do you think it'll progress to a wider range of disorders where the genetics are not quite as straightforward, but where they cause quite a lot of disease burden?
Well, first, I'd like to say I think there's going to be an explosion of gene therapy in the next 5 years. This is a very hot topic. There's been a big backlog of people at the bench showing in animal models that they can basically cure diseases, but because of the perceived dangers and not having the tools properly tested, there's been like a little backlog there. But I think of Mark Fishman's talk on from Novartis on Monday, with your little two-angle square. We know what's wrong in those diseases. We know putting the gene back, there's 10,000 of them, many of them are very approachable. But beyond those, we know a lot from genetic studies about resilience genes, that people can have the same bad gene, let's say in Alzheimer's disease, but a good gene can modify that and make their course much milder. So I think those kinds of genes can be used. And Novartis is also, for instance, trying in terms of deafness, which I think is the most major neurologic disease we all have, to try to get regrowth of hair cells. And I would say, we have the armaments in place now for an initial assault. We have AAV vectors that can be targeted into the brain into different regions. They can even cross the blood-brain barrier. They have a limitation in terms of size, but they're very safe, they're very effective. There's a lot of diseases they can be used for, including the eye diseases we heard about from Eric Pierce. And then there's lentivirus vectors for ex vivo modification of cells that are also shown proven. So I think we've reached a point as a society where people see that these can work. There's a huge number of diseases they can work for, and I think there's going to be more and more pressure from the patients to put these into action.
Thank you.